それでは皆様お待たせいたしました。ではここからは2024年の本表彰受賞者の皆様よりご講演を頂戴いたします。それでははじめにサブサハラアフリカの稲作における小規模農家の気候変動レジリエンス向上のための水管理イノベーションと題しまして、マレーシアのエリオット・ロナルド・ドス・ヨボ様にご講演いただきます。では、ヨボ様よろしくお願いいたします。
Thank you very much. Good morning. It's a great privilege for me to be here this morning, and I would like to thank the selection committees for selecting me as one of the awardees of this prestigious award. My name is Elliott DOSSOU-YOVO. I'm climate change scientist for agriculture, and I work for Africa Rice. I'll make a very short presentation on water management innovations to enhance smallholder farmers' resilience to climate change in rice-based system in sub-Saharan Africa. My presentation will follow this outline. I'll talk a little bit about rice production and climate change in sub-Saharan Africa. And then I'll mention the water management research conducted by my team to improve water control in rain-fed lowlands, to reduce greenhouse gas emission and increase water productivity in irrigated lowlands, to reduce iron toxicity and increase rice yield in irrigated lowlands, and to reduce soil CO2 emission and increase soil carbon budget in rain-fed uplands. I'll briefly talk about the barrier incentive for large-scale adoption of the innovations, and I'll end with conclusion and perspective. Rice has become an important staple food in sub-Saharan Africa, driven by population growth, rising income, and a shift in consumer preference. However, the local production has not been able to keep pace with the consumption, and the gap between production and consumption is met through importation. It is widening and at the cost of billions of dollars for the region. Rice is produced in three major rice growing environments in sub-Saharan Africa: rain-fed uplands, rain-fed lowlands, and irrigated lowlands. The rice yield is low in all the three major rice growing environments, and beside the yield gap, defined as the difference between the potential yields and the farmer's yield, is also large in the three rice growing environments. Poor water management is one of the factors that contribute to the low yields and to the large yield gap. At the same time, rice production in sub-Saharan Africa is largely affected by climate change. The projection indicates that the rice yields will decrease by more than 30% in most of the rice production sites. Also, rice production in sub-Saharan Africa is associated with an increase in greenhouse gas emission. From 1971 to 2021, rice production increased by 0.5 million tons per year, while greenhouse gas emission from rice increased by 0.6 million tons of CO2 equivalent per year. Therefore, there is a need to develop, validate, and scale innovation that improve water use efficiency, rice yield, and reduce greenhouse gas emission in the rice-based systems in sub-Saharan Africa. Together with my team, we developed in the rain-fed lowlands a low-cost approach that is called smart valleys, which help to improve water control in rain-fed lowlands, particularly to enhance farmers' resilience to drought and flooding. It is a participatory approach that is based on careful planning and implementation and based on farmers' knowledge. It requires on average $700 per hectare compared to the classical approach of lowland development, which require about $5,000 per hectare. The Smart Valleys approach comprises three major phases. The first phase is about the selection. The second phase is about the development of the Smart Valleys. And the third phase is about the management. The selection of the inland valley is based on both biophysical and socioeconomic factors. The development of the valleys is based on simplified mapping with the farmers, land development, irrigation, drainage canal developments, bond construction, field leveling. And the third phase, which is the management of the smart valley, has to do with the maintenance of the valley and application of integrated soil fertility management. The smart valleys approach has been now adopted by over 50,000 farmers in 10 African countries. And an analysis of the impact of the smart valleys approach revealed that it more than doubled rice yields in different rice growing environments across different soil properties and management practices. The adopter of the smart valleys approach increased their yields, but also the income by 200 to 1,000 US dollars per hectare, and the food consumption score, which is an indicator of food security, also largely increased. In the irrigated lowlands, as we know, water scarcity is a major constraint for rice cultivation. And at the same time, irrigated rice cultivation is associated with large greenhouse gas emission. So with my team, we evaluated and validated the performance of the alternate wetting and dry approach to reduce the water use and greenhouse gas emission through multi-location trials on farmers' fields. Our results showed that the alternate wetting and dry approach can reduce weed biomass due to the suppression of rain-fed lowland weeds, but also can reduce the water use in rice cultivation, improve water productivity while maintaining rice yield and grain quality. We also showed that in the fields where we have an increase in the soil pH and an application of nitrogen fertilizer that matched with the crop demands, that rice yield was even much more increased in the AWD fields. Our result also showed that AWD significantly reduced the global warming potential of rice cultivation by 30% due to a significant reduction in methane emission. We were also able to develop an integrated approach based on machine learning and soil-water balance to be able to identify the suitable areas for the dissemination of AWD on national scale. We applied this approach to the case of Burkina Faso, and it successfully actually identified where AWD can be implemented at the national scale. Iron toxicity is another major constraint for rice cultivation in irrigated lowlands, and it's affected 440 million hectares in the region. With my team, we developed a mid-season drainage approach to reduce the effect of iron toxicity on rice cultivation. And we validated that approach through multi-location trials on farmers' fields. Our results show that the mid-season drainage approach that we developed actually reduced iron toxicity score by about 40%, reduced water use, increased water productivity while maintaining the yield. The mid-season drainage approach also significantly reduced the global warming potential of rice due to a significant reduction in methane emission. Our results first also show that in the field where we have high soil organic carbon and high iron toxicity score, the mid-season drainage approach was able to increase rice yield compared to continuous flooding. And this is a very interesting result, particularly for farmers whose fields are affected by iron toxicity. In a rain-fed upland, the composition of soil organic carbon actually is a major contributor to soil CO2 emission due to microbial respiration. And this is a major issue in the region because it leads to soil fertility depletion and poor soil holding capacity, making farmers more vulnerable to droughts. So we evaluated the effect of different tillage systems, rice straw management, and nitrogen fertilizer application to increase rice yields, to increase soil carbon budgets, and to increase the nutrient use efficiency in rice cultivation. Our results show that the combination of no-tillage with 3 tons per hectare of rice straw mulch and 60 kilograms per hectare of nitrogen was able to significantly reduce soil CO2 emission, to reduce the soil microbial respiration, to maintain the soil carbon budget, and to significantly increase rice yields and the nitrogen use efficiency. With my team also, we evaluated the barrier and incentive for the large-scale adoption of the water management innovation in sub-Saharan Africa in a different rice growing environments. And we were able actually to identify the key factors that need to be taken into account by government institutions, but also development organizations to be able to incentivize farmers to the adoption of the water management innovations. So in conclusion, I will say that water-related stress leading to drought, flooding, water scarcity, and iron toxicity are one of the major factors that affect rice cultivation in sub-Saharan Africa. And these challenges are exacerbated by climate change. At the same time, rice cultivation in sub-Saharan Africa is now more and more associated with an increase in greenhouse gas emission. So for my team, our research focused on water control in rainfed lowlands, on water management innovation to reduce water use, to reduce greenhouse gas emission, and enhance water productivity in irrigated lowlands, but also to reduce soil CO2 emission and increase soil carbon budgets and rice yield in rainfed upland for sustainability. We also work on the use of GIS and remote sensing and artificial intelligence, particularly machine learning, to be able to identify the suitable domain for the innovation, and we applied it particularly for AWT. We also work on the socioeconomic drivers for the large-scale adoption of the innovation. So as perspective, we plan to address the technical and the environmental research gap in water management in sub-Saharan Africa, particularly now working on the water use optimization in diversification option, where farmers use produce in addition to rice, vegetable, legume, or fish. We also plan to work on landscape water management and governance. We plan to assess the long-term effect of AWD on soil quality and weed infestation, to assess the impact of direct seeding on rice yield, water productivity, and greenhouse gas emission. We also plan to work more on the suitability of the water management innovations, but also on the ecosystem impacts of rice cultivation, particularly paying attention to the local hydrology, biodiversity, and ecosystem services. I would really like to thank you for this opportunity given to me, to the Director General of Africa Rice for nominating me, and you particularly for the warm welcome and allowing me actually to share this few achievements with you. Thank you very much.
ヨボ様、ありがとうございました。それでは続きまして、コロンビアの周辺地域発展に向けたインゲン豆の在来遺伝資源を活用した分子育種研究と題しまして、インドのアンドレス・ハビエル・コルテス・ベラ様にご講演いただきます。それではベラ様、よろしくお願いいたします。
Thank you very much for the opportunity to share this research, as well as for the prize being conferred. Also, thank you to the Agriculture, Forestry, and Fisheries Research Council, as well as the Ministry of Agriculture, Forestry and Fisheries, and the Japan International Research Center for Agricultural Sciences. The work I'm going to present today is about using native diversity of common beans and try to adapt this diversity to regions in Colombia where drought and heat are becoming more common and native communities are suffering. This is a research made by a team that I would like to acknowledge while we are making the presentation, but also to our funders, including the Ministry of Agriculture and Research Development and the Colombian Corporation for Agricultural Research, among other funders. Before going to the topic, I would like to mention that beans, common beans, are a very important cultural heritage in Colombia. Last summer in Japan, this book became very popular again, despite it had been published more than 50 years ago. It's 100 Years of Solitude by Colombian author Gabriel Garcia Marquez. And this is a passage, a quote, where he describes a typical Colombian meal where many people join and gather around the food, including beans. Cooks bump into each other with enormous kettles of soup, pots of meat, large gourds filled with beans, and tots of rice. This describes very well our town, our people, and beans are a very important food and cultural heritage for our communities. Worldwide, also, beans are becoming more popular because they offer a nature-based solution for protein consumption. They are very rich in protein and micronutrients, and therefore they are becoming more popular around the world as vegan markets. This is typically in Colombia, where beans are grown. This is a small village, La Merced, around 2,100 meters above sea level, and there we see how communities live and how beans are grown in very steep hills in the mountains. This area in particular has a lot of rain, but in other places in Colombia, that is not the case. And if we think about bean diversity, diversity in the bean types is as much as in the environments where they grow. This is just a picture showing all the type of beans we have, from dark yellow, red, and different seed coat color patterns and shapes. And they are distributed all across the Americas, from Mexico to Chile, passing by Colombia. Some of them are land races, cultivated varieties, but others are wild beans that might harbor a special adaptation for climate change. This is because those wild beans are being grown by many years in parts of the Americas where rain or drought are more common. And this is just simply a quote from a fascinating book, Darwinian Agriculture, that says natural selection has tested more combinations than humans ever will. And this is why we can learn from wild relatives and land races, because they've been adapted to the places where they grow for many years, even more than cultivated varieties. In the map, in the blue map, we can see the precipitation in the driest period, and some of the beans, like the ones coming from Colombia, are particularly in a region with a lot of humidity and rain, but others might harbor genes and alleles that bring drought resistance or heat resistance. And that's our first focus on the research, and is to build an index that will tell us how much drought tolerance these varieties, these land races and wild relatives might harbor compared to others that are grown in more humid areas. And this also tells us that we can move from raw bioclimatic variables, like just this precipitation index, and we should try to construct and build more complex ecophysiological models, such as this, that are capable to understand the evapotranspiration and the potential for adaptation of these bean varieties and land races. But this is not enough, because these materials are typically grown in the wild, and they are not necessarily adapted to the farmer's field. So the next question is, how can we use this diversity, and how can we bring it into the cultivated gene pool? And to answer this, we've been conducting a type of research that is merging genomic tools with environment variables in order to infer where adaptation is happening in the genome, and try to use these molecular markers to bring adaptation from the wild into the farmer's field. This research has been conducted in collaboration with Matthew Blair at Tennessee State University and with Felipe Lopez at AGROSAVIA as well, as part of his thesis. And what we are seeing here is the 11 common bean chromosomes across the genome, and in each section we are marking a different allele, a different marker, genetic marker, that is predicting adaptation, in this particular case, to drought tolerance. This was done across wild and land races of common beans using more than 20,000 SNP markers across the genome. We also repeated this exercise for heat tolerance, and the map was quite similar to this one, the genetic map. And the important thing to notice here is that there is a lot of determination across the genome responsible for adaptation. This is what is called polygenic genetic basis, and basically tells us there is not one or two genes involved in the adaptation, but there are hundreds and many different genes that are involved in this process. This makes it very difficult for breeders to take this variation into cultivated gene pools, because there is a lot of variation to take care of at the same time. So that's why we need to come up with strategies that will enable a faster deployment of this variation. We also need to understand where this variation comes from, and it basically tells us that it's coming from natural diversity and from the adaptation to a natural gene pool. And for that, I would like to bring the contributions, the theoretical contributions of two Japanese geneticists, Dr. Kimura and Dr. Tajima. Dr. Kimura basically came with the idea that molecular variation across the genome is neutral, more than adapted or more than selected. And based on his hypothesis, most of the markers would be no associated with the adaptation. But Dr. Tajima came with an interesting statistics that tells us when markers are not distributed because of natural variation, because of neutral variation, but instead they are under selection. And basically, based on the Tajima's statistics developed by Dr. Tajima, we see that associated markers, the regions colored across the chromosomes, tend to have a higher score in the Tajima-D statistics. And for geneticists, this basically tells us that adaptation is not just polygenic, but it's also under divergent selection. That is happening across the genome because different forces are co-occurring in these regions. In practical terms, not so much in Tajima's statistics, it basically tells us that adaptation to draw and heat is good under stress, but not necessarily when conditions are ideal. This is basically a trade-off for for varieties that are deployed into farmers' field. So based on this risk, polygenic adaptation, and the trade-off that we are detecting here, we need to come up with a strategy to bring this adaptation into the cultivated varieties. And the strategy that we are presenting here is hybrid beans, making a hybrid between the cultivars and the wild beans. And this donor of draw and heat resistant looks like this. It's a Phaseolus species related with common bean. It's tepary bean, Phaseolus acutifolius, grows in Mexico. And this is typically a place where it grows, in very arid and dry soil. So by making crosses, hybrid crosses between this wild relative and the beans, we can determine which of these regions are useful for the adaptation and how we can predict that adaptation. And this is basically what we are looking at that here at the two different stations in Colombia by the Caribbean coast, where there is a lot of heat and draw. And this is the predictability of the two different models for 5 different traits, from overall yield, number of pods, number of seeds, biomass of seeds, and vegetative biomass. And interestingly, with the molecular markers that we detect, despite the polygenic architecture, we are able to predict up to 75% of the adaptive response of these hybrids. This opens the door for using this genomic prediction in hybrid breeding to bring this variation from wild and land-raised gene pools into the cultivated varieties more efficiently, despite the polygenic architecture and the environmental trade-off of the materials. This is also extended to different types of markers, and interestingly, despite we work with more than 20,000 SNP markers, genetic markers across the genome, we don't need that many variation. Around 250 would be enough to achieve 75% of the predictive ability. That again tells us about the polygenic architecture. There is no 1, 2, or 3 genes, but instead there are hundreds of genes involved in this response. But this type of models, genomic prediction in hybrid breeding, are capable to predict the polygenic adaptation in more complex scenarios. This first example has been tried across 87 hybrids between the common bean and the Phaseolus tepary bean. but this needs to be expanded as part of further research. But moving again out from the comfort zone, which is science, we need to start thinking back into what farmers need and what is really useful for them. So these models enable us to select the varieties that bring the adapted alleles, the adapted variation from the wild, but we still need to multiply these seeds and deliver them to the farmers. And that's the second part of the achievement. And it's not just to make science, but also make this science and the varieties reachable to the producers. And this involves adaptation and yield trials of the materials that have been selected, plus a seed multiplication scheme that ensures that farmers can assess this variation. And this is just an example of the farmers working in the field with some of the varieties we tried. A great part of this job was achieved thanks to our long-term collaborator, Diana Peláez, and also to the farmers who have been selected through participatory breeding, the varieties more useful to them. And part of the selection was in this yellow bin. This is the commercial type, and this is the type they've selected through participatory breeding after the adaptation and yield trials. And this one, the ones they selected, is the one that we multiplied and delivered to the families, almost 40 families and more than 200 kilograms of seeds selected and specially adapted, delivered to the farmers. With that, I would like to bring these conclusions. And first is that beans are a main source of dietary protein and macronutrients, and therefore they offer a nature-based solution for food security in vulnerable communities. We bring and present different genomic strategies that, when coupled with ecological variation naturally happening in the gene pools, and with some statistics proper from the genetics field, we are enabled to explore and predict the basis of adaptation and how these relate with the stability across the environments. And what we managed to learn is that hybrids between common bean and temporary bean exhibit draw and heat adaptation, but this adaptation is polygenic and is under a divergent selection trade-off. And this is why it's very challenging to utilize this variation into varieties. But we came up with this strategy, which is to predict the potential of abiotic strains using genomic prediction in hybrid breeding, with achievements around 75% of the predictive ability. And finally, an important remark is not to stay just with this scientific prediction, but to move into the farmers' field, and participatory breeding enables this strategy where farmers can select by themselves the best varieties after adaptation and yield trials, and this can be coupled with uniformity and stability tests as part of the seed multiplication chain. And at the end, this will empower local communities as long as they keep producing their varieties based on the learnings from this participatory breeding strategy. With that, I would like to, again, thank you very much for this prize and opportunity to share the research, and welcome to Colombia and to eat more beans. Thank you.
ベラ様、ありがとうございました。それでは続きまして、革新的な細菌層解析技術における飼料からのフードチェーンおよび農産加工品の安全性と栄養の向上と題しまして、タイのジュタマット・クリンソーダー様にご講演いただきます。では、クリンソーダー様、よろしくお願いいたします。
Good morning. Thank you so much. Good morning, our distinguished guests, ladies, gentlemen. My name is Jutamat KLINSODA from Thailand. It's honor to be here and to be a part of your prize giving ceremony. And I'm very grateful for this opportunity to share my research. And since 2020, I hold a doctorate degree in nutritional science, and after that I have conduct solo research in food safety and microbiome. and I also have experience in probiotic and prebiotic. Sorry. Okay. And for the cooperation with Japan, I developed food safety training for Bangladesh Food Safety Authority, help them to implement and learning a food control system in Thailand. This training was supported by JICA. And 2015, I joined the JIRCAS project. This project we developed of a functionalถั่วเน่า and this project we distribute theถั่วเน่า culture or natto in Thailand to the north of Thailand to because we have the bacteria culture that have functional food. And I also teaching international food law and do training about food law and probiotic research for several ASEAN countries. I also have been invited from several journals to be a reviewer this year. For my expertise, 16S RNA sequencing is a key methodology for multidisciplinary research activity for me. 16S RNA genes has been extremely well conserved, and this gene specific for bacteria species. Therefore, if we would like to exploring bacterial microbiome, we can extract DNA and sequence on 16S RNA genes, followed by bioinformatics. And then we can generate the profile of bacterial population in samples. This is a benefit more than culture method. Conventional method, because the 16S RNA genes with microbiome data, we can explore the whole population, include the one that not culturable. And I have been applied 16S microbiome technology in several fields such as feed safety and animal nutrition in pigs, food safety and quality and nutrition, and also investigation of earth and plant abstracts, because the microbiome data is useful for understanding the microbiome and immune system in animal and also in humans, especially in human intestine, about diet and gut interactions. And for the first project, we developed the new pig feed products by soaking lactic cereal in lactic acids, and also add phytase supplementation in cereal, because phytase phosphorus is important for bacterial growth and also bacterial metabolism. We hypothesize that the more phosphorus availability and substrate availability in the upper gut, if they're more absorbed in the upper gut, it will have less phosphorus to support the growth of bacteria or enteric pathogen in the lower gut, such as in large intestine or feces. This project, we found that it could improve phosphorus availability and absorption in the pig gut, in the upper gut. And also we could find the change of fecal microbiome and the expression of barrier function gene related to immune genes. Our result, we found that feeding the lactic acid treated cereal and phytase supplementation improve animal performance and improve animal gut health, and this could reduce fecal contamination, and this can enhance meat quality. And also this project, we could reduce the fecal contamination that can prevent the transmission of zoonotic disease into human. And after that, we also from our previous study, we hypothesized that the pathogen may originate from pork on pork may originate from the feces and edible pork organ. Therefore, we correct all pig's organ for consumption and bacterial on the hands of the worker at slaughterhouse, and we monitoring pigs one by one. So we have in total 12 pigs. And then in this study, we aim to develop microbiome database because in Thailand, people like to eat uncooked edible pork organ and also some fresh products. Therefore, if we have a microbiome database, we could provide some recommendation for consumer that consume edible pork organ without cooking, and this could reduce the mortality rate for Streptococcus suis infection in Thailand. And here is the microbiome database. We could find the relationship and composition and similarity between microbiome on edible pork organ and human hands. And if you have a look on the picture, we could see that some organ like a Thomson, because we separate some. Because Thomson is under the head, so this sample was sampling separate, but other sample we sampling together because it's connected to the gut. Therefore, we found the cluster together with between the edible pork organ. So this information we could conclude that the hygiene during carcass processing is potential step for cross contaminations. Another projects, I also apply a microbiome for study food quality. For we have the new plant-based protein such as mung bean and duckweed. And this one we want to add to the plant-based meat protein or meat analogs, which is popular this year for plant-based meat. So we monitoring the change of microbial profile, metabolite during storage, and also we connect the metabolites that the bacteria produce during storage with a sensory evaluation to connect with how consumer accept it. And this product we could provide like some functional food information for alternative plant-based protein. Here is the result of the plant-based protein products. We could found that the microbiome database can use to monitoring the bacterial profile in raw ingredients, because the conventional method that some bacteria is not grow on the agar, but for the microbiome database with 16S and A genes, we could identify the whole population like cyanobacteria that is may contaminate from the water that cleaning soybean. And also we could use microbiome database for monitoring pathogens, the growth of spoilage bacteria during storage, and also shelf life prediction by analyze timing dependent change in abundance of certain bacteria. If you have a look, we could use bioinformatics and RStudio to find the interaction and relationship between sample and abundance of bacteria in each ingredient, and also the growth of bacteria and chain during storage. Another project, I also investigate the Thai herb and algae as plant-based functional ingredients, because in Thailand we have like several we have high biodiversity of plant-based protein and Thai herb and algae. So we hypothesize that the complex carbohydrates in herb and algae are prebiotics Prebiotic is one that can support the growth of bacteria in our gut. So therefore, we assess the prebiotic property and evaluate the effects of Thai earth and algae on fecal microbiome from the donor under human gut models. For this gut model, they are like a fermenter that we incubate the feces from the donor to find the relationship between diet and gut interaction and how the Thai earth and algae modulates gut. And also these projects, we could use the information for develop like functional prebiotic ingredients with help claim how diet affects on human gut. And this can also enhance the value of Thai earth and algae. So for the overall, you could see that the microbiome translation research can be applied from agriculture farms such as feed development or study the animal gut health, and also can study like for food production to monitoring food quality assurance, and also study the functional food, and also investigate on the personalized nutrition recommendation on human gut models. And the microbiome can apply for public health study as microbiome database. It can explore the whole bacteria or microbiome in gut. Therefore, it can help for future pandemics that we can use microbiome to monitoring. So lastly, for my achievement, I thank for all funding and the director and co-investigator from Institute of Food Research and Product Development, Kasetsart University, and also other organizations in Thailand. And thank for JCAS for a long collaboration with Institute of Food Research and Product Development, Kasetsart University. And lastly, thanks my PhD supervisor and my colleagues as co-author of PhD project as veterinary medicine, Vienna, Austria. Because we produce currently more than 10 publications during 4 years. And thank you so much for having me at this ceremony, and I look forward for the future cooperation and investigation in food and nutrition area between Thai and Japan. And thank you for your attention. For the further interest in my publication, you can search on the internet. Thank you so much.
クリンソーダ様、ありがとうございました。以上、3名の受賞者の皆様から講演を頂戴いたしました。皆様どうぞ今一度受賞された皆様に大きな拍手をお送りください。それでは以上をもちまして2024年若手外国人農林水産研究者表彰を終了いたします。会場参加の皆様にはこの後少々ご案内がございますので、どうぞ今しばらくお付き合いくださいませ。本日はご参加またはオンラインでのご視聴誠にありがとうございました。